Every expectant mother knows that what she eats impacts her baby. And now research shows that is also true for our cows. Maternal consumption of ReaShure during late gestation had a positive effect on the in utero calf, setting her up for better health and potentially even higher milk production once she joins the milking string. Learn more at balchemanh.com/launch and launch your herd for life.
Well, I'm very sad I couldn't be there back in upstate New York. I haven't actually been back since I finished my PhD, but I am fortunate to still be joining you guys today here. And I just wanted to, with my talk, give a little bit of there's a bit more background. Dr. Decal really went through some really awesome work that's being done in the dairy cattle industry. I wanted to take a step back and just explain the broad reasons why we think about diet and why it might modify the epigenetic state, and then also kind of go into some specific examples within the rodent and human literature about that we've contributed to related to dietary methyl donors. But just so everybody is using the same terminology, I always like to take a step back and make sure that when we're talking about genetics, this is really coming from the Greek word geneticos, or origin. And the term was really popularized by Bateson, who was a proponent of Mendel's work with pea plants, where he was looking at the heredity of traits. So it's really about the study of the transmission of that DNA sequence and how is it inherited across generations of different organisms. And just to remind everybody, DNA is composed of the nucleotides adenine, thymine, cytosine, and guanine. And really remember that cytosine there, because that's what gets methylated as one of our epigenetic marks. Epigenetics, however, epi really refers to over or on top of or above. So it's above the genome in a sense. And it was a term that was defined by Waddington in 1942 as any change in phenotype that occurs without changes in genotype. So it's very quite broad there. But the field really now focuses on studying changes in gene expression without changes in DNA sequence. And people use the term epigenetics a bit differently. I think initially it was meant to be focusing on marks in the genome that are mitotically heritable, although some folks just use it to mean anything that's a change in gene expression. And I think going back to that original Waddington definition, we kind of allow for that flexibility. But the questions that drive the field of epigenetics, and then some of what Dr. Decal got into, are why do cells and tissues look and behave differently despite having the same DNA? And why do genetic risk factors, when you look in big genome-wide association studies, looking at diseases and other phenotypes like height, they really only explain a fraction of the variation for most individuals? And then why do factors such as birth weight strongly and independently predict chronic disease? Some of you might be familiar with the idea of fetal programming, or what was originally called the Barker hypothesis, which started in humans with Barker noting that birth weight in the UK was an independent predictor of coronary heart disease. And, you can take into account cholesterol and smoking and whatnot, but an individual's birth weight still strongly predicted. So suggesting that something that happened in the intrauterine environment was linking to disease risk decades later. There's, of course, other questions that drive the field of epigenetics, but these are some of the basic things that scientists have thought about and said, okay, independent of our DNA sequence, why do we see these observations? Just to remind folks, your DNA gets expressed, it gets transcribed by RNA polymerases, and then it ultimately gets translated and turned into proteins within the ribosome. But this sort of picture that you see in a classic textbook is a little bit of a naive take on the true complexity of the genome. And the genome is really structured, I think we all know about chromosomes, but really you get down into these microunits of chromatin fibers and then individual nucleosomes, which are the DNA strand that is wrapped around histone proteins. So you have this very complex wrapping of DNA, and then this three-dimensional structure where you can have broad interactions of DNA sequences that if you were to lie them out straight, they would appear quite distal, but they're physically interacting in the genome. And this allows for a lot of complexity in how DNA is ultimately transcribed, regulated, and what proteins are made. So chromatin needs to be accessible by a variety of transcription factors that recruit RNA polymerases to drive transcription. modifications of the DNA can really affect how accessible that DNA is. So those major modifications that we think of in terms of epigenetics, and these modifications grow, and there's big debates about whether they're true epigenetic marks, but the classical ones that we think of are DNA methylation, where the cytosines are methylated, or histone proteins themselves being methylated or acetylated. And what basically happens is that can lead to chromatin being super condensed and inaccessible to different activators of transcription and leading to more recruitment of repressors of transcription, or it can lead to this more open state where the chromatin is decondensed and you can have large protein transcription factor complexes come in, recruit RNA polymerases, and turn on genes. DNA methylation is typically found at cytosines followed by guanines. So you might hear it referred to as CpG methylation. And histones are a family of proteins where it's quite complex, the naming system, but their lysines are typically post-translationally modified by acetyl groups or methyl groups, and lysines amino acid code as K. So you'll see things like H3, which is histone 3, K27, so the 27th lysine, and then acetylated, or you might have Me3, which is trimethylated, where there's 3 methyl groups. So if you come into the epigenetics literature, you'll see this sort of word soup, and it only gets worse when you start to look at all the readers and writers and erasers of epigenetic marks. And so just as an example, the promoter region of genes are commonly where transcription factors dock and turn on gene transcription. And so you might have a repressive mark on the histone itself or a 5-methyl cytosine. So that DNA methylation is associated typically with this inaccessible chromatin, inactive promoters, and genes being turned off, although that's not universally true. And then repressive histone marks are commonly methylated as well, like H3K9 trimethylation or H3K27 trimethylation. Whereas accessible chromatin like H3K27 acetylation allows for this open state of chromatin and for gene transcription to occur. So these little marks link the physical structure of how the DNA is wrapped around its histone proteins and then how accessible it is and how transcribable the DNA is. And I want to just put a quick note about how we characterize the epigenome. So it's really high throughput DNA sequencing that has facilitated discovery within this field. And so we typically look at DNA methylation with whole genome bisulfite sequencing, where you treat the DNA with bisulfite first. It converts cytosines to uracils, but methylated cytosines are protected from this. And so when you sequence the DNA, the cytosines that you see there are actually the methylated cytosines, and then the uracil, which will be read as a thymine, comes out as thymine. And we also do chromatin immunoprecipitation, where you cross-link your DNA and your histones with something like formaldehyde, and then you use antibodies specific to those histone marks. So an H3K27 specific, acetylated specific antibody, and you pull down that specific mark, and then you do some cleanup and sequence the DNA, and then a huge bioinformatic approach to try and determine what DNA sequences what your sequencing actually aligns to in the genome, and then you can make inferences about what marks were specifically there. So where does nutrition come in? Well, part of it is that it's the raw materials of epigenetics. So we've been talking a lot about methylation and acetylation. Well, diet is the major source of methyl donors to generate the S-adenosylmethionine, which is the universal methyl donor that all methyltransferases in the cell use. So there's a whole family of hundreds of methyltransferases, of which the DNA methyltransferases and the histone methyltransferases are just a subclass of them, but they use S-adenosylmethionine, or SAM. And that looks here basically like this, what we call the methionine cycle. So you have methionine that is activated to SAM. SAM can donate its methyl groups and form some sort of methylated product, whatever the product of that methyltransferase is. You generate SA, which can be cleaved to homocysteine, and then homocysteine has to be remethylated by methyl groups coming in from the diet, commonly folate with the help of B12, or with serine, or with choline or betaine. And you reform methionine, and then it becomes this big cycle. And so you can imagine if you have a change in the dietary methyl donor availability, you will eventually have a change in the SAM levels and the methylation potential of that specific cell type. You also have the acetyl groups coming from the oxidation of dietary macronutrients predominantly. And this allows us to link bioenergetic state and mitochondrial integrity, where the acetyl groups are really being formed, to changes in chromatin accessibility and gene expression. And just the number of hypotheses you could come up here about factors in the diet that change the amount of acetyl-CoA or acetate that is being produced that could then turn on and off genes is seemingly endless. And that's why we have these big high throughput methods to measure what's happening in the entire genome. But just for those who want a reminder, you are forming acetyl-CoA either from fatty acid oxidation or glucose oxidation. It enters the tricarboxylic acid cycle as condensating with oxalo acetate to form citrate. And then it's sort of this cyclical loop. But that acetyl-CoA can be exported in the cytoplasm through the help of an enzymatic reaction called ACLY, or acetyl-CoA lyase. And then that acetyl-CoA can actually form as the substrate for histone acetyltransferases to acetylate histones and change the conformation of DNA and typically activate DNA for transcription. This picture also nicely notes that we're still figuring out all of the things that regulate histone acetylation, the things that place the marks there, and then the things that remove those acetyl marks, and same for methylation. And a lot of intermediates within the tricarboxylic acid cycle, or TCA cycle, are regulators of these processes. There are other dietary components too that are also influencing what we call the readers, writers, and erasers of these different epigenetic marks. So the things that place the marks there, the things that are actually recognizing the marks, and then the things that remove the marks. So for example, the 1011 translocation or TET enzymes oxidize our cytosine methylation or DNA methylation, which is a major active step in the demethylation process. So in theory, turning a gene on, and that requires ascorbic acid or vitamin C as a major cofactor. So you can start to think of hypotheses about changing dietary vitamin C, and does that change the activity of a TET enzyme? And then subsequently change DNA methylation state and gene expression. The histone deacetylases are also inhibited by soy isoflavones, and genistein is the most well studied in this case, where they have been shown to, particularly in cancer cell lines, increase histone acetylation and reduce DNA methylation. And many cancers have hypermethylation, so there's a lot of interest in how soy isoflavones might have anti-cancer properties in humans. It's mostly in vitro data that we have now. So don't want to get too excited, but it's an interesting basic science point where these seem to cooperate with histone deacetylases, seem to inhibit histone deacetylases. And you might be familiar with phytates. They're commonly thought of as an anti-nutrient or a bad thing. In human nutrition, we think of this way, and I think in agricultural nutrition, it's pretty much the same, because they can bind cations, particularly iron, and reduce their bioavailability. But phytate has been recently described as a histone deacetylase activator, suggesting that there's a link between this thing that we've always thought of as being bad and histone acetylation, which would, in theory, influence gene expression. And this was a rather recent paper, but opens up a door for this thing that we've been trying to remove from crops might have having had some function. But my talk today, I'm going to mainly focus on dietary methyl donors and really hone in on choline, because this is where most of the work has been done as far as diet goes, and clearly causally changing the epigenetic state. There's certainly a lot of studies of obesity models and things like that, and looking across different tissues and looking at changes in the epigenetic state, but really what the different mediators of those changes in epigenetic state are not clearly known, whereas the methyl donor availability is quite proximal to the epigenetic changes. And so I just sort of highlighted the methyl groups there. And the thing to note here is that choline, which is ultimately oxidized to betaine to partake in methyl donor transfer, and you can eat betaine in the diet as well, comes with 3 methyl groups. So it's quite a bang for your buck as far as eating these sorts of methylated nutrients goes. And we also eat quite high quantities of it. Both choline and betaine we're eating in milligram amounts, compared to something like folate that we typically think of with the methionine cycle and remethylating homocysteine to methionine. That's only in microgram amounts. So choline is both quantity-wise and just the number of methyl groups that it comes with within an individual unit is quite a large methyl donor in the diet. And so it's a trimethylethanol ammonium compound. We often see it grouped with B vitamins. And in humans, it was deemed an essential nutrient in 1998. If you look at the NRC's guidelines, there's different studies of different animal models need or animal species need different amounts of choline and or betaine. We just talked about cows, and I know they have a lot of foregut fermentation that degrades the choline, and betaine might be more important there. But choline itself in humans is quite important. And when you deprive it from the diet, you get fatty liver phenotype and elevations in liver functional tests like ALT and AST. And most of the data that informed this dietary recommendation was a controlled feeding study where you deprived choline from the diet and then added it back in. And it was really only done in men. And there was also studies from individuals undergoing long-term total parenteral nutrition, which has no choline in it. So they're essentially a special subset of the population that typically have some sort of intestinal dysfunction that they're not able to take food in enterally, but they serve as a sort of model of choline deficiency. And they have a fatty liver phenotype as well. But from these two types of studies, adequate intake values were established for all life stages. So for pregnant women, for example, the choline intakes were derived from studies in men. So there's a lot of research to be done in dietary choline. This is just a basic overview of the metabolism, and most of us know about choline because of this upper pathway here where choline gets phosphorylated, condensed with DAG after activation to CDP-choline to produce phosphatidylcholine. But choline can also enter the mitochondria and undergo oxidation to betaine, where betaine can then, through the activity of betaine hydroxymethyltransferase, remethylate homocysteine to methionine and reform our S-adenosylmethionine that can then be used as a methyl donor. And I just want to call out that there's a second pathway to make phosphatidylcholine in the liver where you can trimethylate a phosphatidylethynolamine to form a phosphatidylcholine, and that'll be relevant elsewhere. But the methyl groups, after betaine donates one of its methyl groups to remethylate homocysteine to methionine, that's only one methyl group. And so we know choline comes with 3. Those additional methyl groups can go back into the mitochondria as dimethylglycine and then be donated to and enter the folate-mediated 1-carbon metabolism cycle and be utilized that way. So there's this broad interconnection of dietary choline, dietary betaine, dietary methionine, dietary folate, all to maintain what we call the 1-carbon nutrient status of the cell. So when we think about how choline affects health, as I said, that it's when you deprive it from the diets in humans, you get a fatty liver phenotype. And that's really because phosphatidylcholine is required to export hepatic lipid. And so when you don't have choline, the first thing you see is really a defect in the amount of phosphatidylcholine that can be produced in the liver and then liver fat buildup. We also know choline is rapidly acetylated in neuronal tissue, and also there's some non-neuronal cholinergic systems. And some of the actions of choline in health might be mediated by acetylcholine. There's also hundreds of methyltransferases that are using its methyl groups. So anytime you dump choline into the system, into a biological system, you are providing methyl groups that might be driving any number of pathways. And it's really hard to simultaneously measure all this. And then linking that is choline influencing the epigenetic state and really teasing apart mechanistically how choline influences health and whether it's simply due to changes in epigenetic state are quite difficult. And we're still in much of a discovery phase within the field. And I think Dr. Deco alluded to this a good bit too, of, in cows, you might only have blood, which is the readily accessible tissue, and rats and experimental mice and laboratory animals in general. We have this big question of, you could feed 3, 4, 5 different doses of choline in the diet and then sacrifice and take all their tissue. And you could do this in the context of pregnancy, you could do it in non-pregnancy, you could do it in cancer. There's just an infinite number of contexts in which we could say, how does choline intake influence the epigenome and what is it doing to overall health? And we really don't know what sites in the genome, what genes, where their epigenetic state is actually really pliable and sensitive to dietary methyl donors. So that's all an active area of research, but that doesn't mean we haven't studied it. And there's good reason that we have focused a lot on choline in the context of pregnancy. and not just because we have very little data in pregnancy and the pregnancy DRIs are derived from studies in men, but we think that particularly tissues early on in life are more sensitive to variations in the dietary methyl donor supply. The epigenetic state of cell types and which will ultimately go to be tissues and specific tissue niches, they are more sensitive early in life. And so some sort of disturbance to the epigenome early on might have broader consequences than later on in life. And some sort of disturbance in the methyl donor availability at the time when the DNA methylation state, for example, is being established might have long-lasting impacts that are not necessarily able to be recuperated by supplementing with choline later on in life. And we also know that pregnancy is a physiological state of methyl donor stress. There's a lot of methyl donors that are required, or methyl groups, and choline itself is just required to build an entire baby. You have a lot of phosphatidylcholine going into building all those cell membranes. You have a lot of acetylcholine that needs to do signaling to promote neuronal development. And so choline is just a time of high stress. Part of that is because choline in the amniotic fluid and fetal cord blood are several-fold higher concentrations than what you see in the maternal compartment. So the placenta is actively pulling choline from the maternal plasma and concentrating it within the fetal compartment to facilitate the building of the fetus. And the rat hepatic concentrations of choline in dams, so in the mom, are significantly diminished during pregnancy. So at day 18, you can see that several of the choline metabolites, and so this study here actually nicely did female non-pregnant mice, and then female pregnant mice fed a deficient diet, then pregnant mice, and then pregnant mice fed a deficient diet. And what we see across a lot of the metabolites is that the pregnant mice, their hepatic choline metabolome that was targeted here looks a lot like the female deficient mice. So phosphocholine, you can see that the female deficient looks quite like the pregnant. So you're depleting some of these choline metabolites just naturally with pregnancy. And once you get to the very end of a rat pregnancy, which is about around day 21, it can go as long as 23 days, you see a dramatic drop in the liver concentrations of choline relative to the non-pregnant female. So during those last few days of pregnancy prior to parturition, you're getting a severe depletion of choline within the liver. And that's really to facilitate the plasma having plenty of choline that then the placenta can uptake it and concentrate it in the fetal compartment. So there's a choline stress in general, and then with that, a methyl donor stress overall. DAMS-fed choline deficient diets have a lot of consequences for fetal brain development. There's impaired cortex and hippocampal development. Their overall brains are small. When you stain for all sorts of factors related to neurons and neurogenesis, you see reductions in them. They have worse visual spatial memory and auditory memory relative to just animals fed controlled diets. But interestingly, when you supplement even a regular control amount of choline, you get lifelong improvements. And so what you see here is animals that are the offspring who either got the typical control chow amounts, which is about 1 gram per kilogram in a mouse chow, or about 4 times that, and at 2.5 months of age, and then aged all the way out to 2 years, which is quite old for a mouse, or sorry, a rat. In this study, this was a rat. They were doing Morse water maze tests, and basically all you need to see here is that they're making very few errors, and whereas the rats fed the control chow, their errors are increasing as they age. So there's that age-related memory decline. And then those that got the supplemental choline during gestation only don't really see an age-related decline, and they see fewer errors at all the different time points. And this is really linked to improvements in their memory, particularly visual spatial memory. In addition to neurons being impacted, and this is where we're going to start to get to some of the actual epigenetic stuff, the endothelial cell populations in the hippocampus, so you need an adequate amount of blood supply to the neurons to be able to supply them with oxygen and other nutrients, not just neurons, but glia and other cell types as well. When you feed choline deficient diets, there are reductions in endothelial cell proliferation and reductions in the total number of blood vessels. And interestingly, there's decreased DNA methylation of ANGPT2 and VEGF-C. And this is an example here where you're looking at specific CPG islands in choline deficient, normal choline or choline supplemented animals. And you just see this loss of DNA methylation at VEGF-C, which is a primary pro-angiogenic factor, which this loss is associated with an increase in VEGF-C. And we think that this is probably either causing early differentiation of endothelial cell types from their progenitors, which is why you see a decrease in the overall number, or this is a compensatory response where the cell is trying to increase as much VEGF as it can to promote the angiogenesis because something else about choline deficiency is compromising it. There's this really classic experiment, and I'm glad Dr. Deckel didn't talk about it, but there's the agouti mouse. When you supplement with methyl donors in the diet, including choline, you can actually drive the coat color. And so they have a transposable element within their genome that is able to be modified by a choline supplementation. And it's just this very nice readout phenotype, you could say, where you supplement moms with choline and folate and B12, and the normal wild-type mouse has a yellow coat, and then when you supplement, you get this brown coat color, what's called Pseudogoody. And it's a very specific thing to this inbred mouse strain, but it's nevertheless sort of highlights the power and long-lasting impact that an early nutritional intervention during pregnancy can have. And this is, I should say, is associated with the percentage of cells that exhibit methylation in this specific transposable insertion within the gene, the AVY gene here. So you see a greater proportion of supplemented mice are showing higher levels of methylation, and then you get from that additional methylation, you're getting more of a pseudo-agouti phenotype. So simple addition of methyl donors to the dam diet can shift coat color, and there's some other obesogenic phenotypes that you can see. So what about in humans, though? And I know I only have like 7 minutes left, so I'm going to go through some of the work that we did. The Caudill lab, where I trained with at Cornell, some of you may know Marie Caudill, she conducted, prior to my time entering the lab, although I've worked with this data a good bit, that like 12-week controlled feeding study in third trimester pregnant women. And so the two levels of choline that were fed were 480 milligrams a day, which is about the recommended intake. Notably, habitual diets contain about 100 to 150 milligrams less choline than this, and then supplemented with an additional 450 milligrams of choline a day. And this was in the context of an otherwise nutritionally replete diet. So we weren't doing anything sort of, we weren't, their folate intakes, B12 intakes, everything else was relatively optimal, or even like super physiological, very high intakes. So all the effects that we're seeing are not due to choline sort of compensating for another deficiency phenotype. But we published that we see plasma choline levels going up over time. So there's a normal increase as the maternal hepatic stores are mobilized to allow the placentas to concentrate that choline in the fetal compartment. But adding additional choline increases the availability of choline to the placenta, but also to other maternal tissues. We're seeing increased betaine as well, suggesting that plasma choline, or that choline that's being eaten is oxidized to betaine, which is capable of participating in 1-carbon metabolism. And then the product of 1-betaine does donate a methyl group is dimethylglycine. We're seeing that go up as well. So we think that supplemental choline is driving additional methylation reactions. And sort of the most interesting phenotype that we care about, at least clinically, is that supplemental choline improved the mean reaction time in visually guided reactive cycads. So it's really just a test of if you introduce a new item in the visual sphere, but sort of peripherally of an infant across the first year of life. So there are different time points tested at like 4, 7, and basically 12 months of age. The reaction time is the number of seconds it takes for the infant to recognize that there's something new in the peripheral vision and for the fovea of the eye to actually target it. And you can see the choline supplemented infants came out better, so they had a lower mean reaction time. So their eyes were adjusting faster to the new item within the peripheral vision. And notably, this test wasn't calibrated as they age. So everybody's getting better over time, as you would expect, as there's cognitive development across the first year of life. But the choline group persisted. And Unfortunately, there's not a paper yet, but these children were followed up to 7 years of age, and some of the cognitive tests, the choline supplemented group was still performing better. So look out for that publication in the future. And really getting to some more epigenetics, we don't really know if that cognitive phenotype is due to some change in epigenetic state, and there likely would be changes in epigenetic state if you could access fetal brain or something like that, but we don't see that necessarily. We have to rely on animal models for that. But what we did see is in plasma, cortisol was notably lower in the sort of metabolomic assay that we did. And in the higher choline supplemented group, the cord plasma, so this is what the fetus was in theory exposed to, was reduced. And so just to remind folks of sort of the glucocorticoid pathways, you have corticotropin releasing hormone that can be made in the hypothalamus or the placenta that is then going to the pituitary to release adrenocorticotropin hormone, and then, or ACTH, and then you have cortisol being produced ultimately at the end of the day, and that ends up in plasma. And what we saw was in the placenta, you see the CRH gene becomes hypermethylated, and then the actual gene expression in the placenta is reduced, suggesting that the additional choline increased DNA methylation within the placenta, and that had a consequence for this, the glucocorticoid system in general and cortisol production at the end of the day. And really, we don't know what the consequences necessarily will be of having seen less cortisol in vivo, but it could be programming the hypothalamic pituitary axis and stress reactivity in the future. And that's something that we'd have to follow up the kids even longer. But it's nevertheless an enticing, very targeted example of how choline supplementation influenced the epigenome. And when you look in the cohort broadly, both in maternal leukocytes and cord blood leukocytes, their overall global DNA methylation wasn't really significantly altered, but the placentas was. And this is, this is not site specific. This is looking at the entire genome, and it's kind of interesting. It suggests that the placenta is uniquely susceptible and sensitive to the dietary choline supply. For decades, we kind of just thought of, mom mobilizes choline, and then you get more in the fetal compartment, and the placenta is just transferring it. But it seems like additional choline is actually impacting the placenta. And we saw the same with histone marks as well. So H3K9 dimethylation was specifically up as well. And the gene expression of the enzymes, the EHMT2 and DNMT1 that place those marks was increased. So the placenta was sensitive to the dietary methyl donor supply and changed the epigenetic state in response to choline. We also have ongoing now, this is a teaser of our current study where the publications are under wraps, and this is really the cohort that I started. This was a free living randomized controlled trial, and individuals were supplemented with just 25 milligrams per day on top of their habitual intakes, or 550 milligrams of choline per day. And as I said before, the controlled feeding trial was really the control arm, or the lower intake arm, was about 100 to 150 milligrams higher on average than what typical intakes are. So it likely limited our ability to see the true choline stress of what the everyday mom is going through. And so we assessed them at baseline at gestation weeks 12 to 16. So before, where it was a third trimester trial, we went back into the second trimester here, and then followed them longitudinally. And we were really interested in the availability of methyl groups for this enzyme PEMT that converts phosphatidylethylamine to phosphatidylcholine. And those PC species are enriched with the omega-3 fatty acid DHA. And we thought that the low choline intake might compromise the activity of this PEMT enzyme. And I know we're about out of time here. So just to show you the results, we see that choline supplementation in the pregnant women across the third trimester, we're increasing the availability of phosphatidylcholines enriched in DHA and plasma, and that the red blood cell DHA content, which is a surrogate marker of what's happening in tissues, is dramatically increasing, sort of the equivalent of adding an additional DHA supplement on top of what they're already eating, but it's just choline making the availability better. And we also see that choline and betaine and dimethylglycine are going up, but it's a bit more dramatic than our last study, as we would expect, going longer and also having a control arm with lower intakes, and isotopic enrichments, because we gave a tracer, show that PEMT activity is increased. And so we're really going on now to do ChIP-seq to look at histone state, whole genome bisulfite sequencing for DNA methylation, and RNA-seq to take this broad systems biology approach to see how this choline supplementation in the context of normal dietary choline intakes influence epigenetic state, and also do some additional infant cognitive phenotyping. We can talk about this more at the panel, but there's a lot of challenges for human nutrition in integrating epigenetics.
At Balchem Animal Nutrition and Health, we strive every day to deliver results you can see in your animal's productivity and your bottom line. From a smooth transition into the milking string for your fresh cows, to a happy welcome home from your furry friend. From a strong start in your poultry flock, to consistent weight gains for your finishing hogs. We expect to earn your business and your trust with our people, our products, and our science. Our people have an intense passion for your animals and your success. You can count on us for honest, candid advice and practical solutions for your toughest challenges. As the global leader in choline production, chelation, and encapsulation technology, we take our obligation to you and to the environment seriously. Our products are backed by the most extensive and thorough research portfolio, while our business is committed to advancing environmental sustainability and animal welfare. In the end, it all comes down to results. Balchem delivers real results you can count on, results that exceed your expectations, and results that bring true value to your bottom line. Leading the charge to meet the nutritional needs of ruminants, monogastrics, and companion animals, Balchem offers a growing portfolio of nutritional products and a dedication to innovation and industry sustainability. Balchem is here to solve today and shape tomorrow.